So thank you. Yeah. Hi everyone. I'm Sam. I'm a postdoc in Guy Pacino's group. We are now in the Human Technopole, which is a brand new research institute in Milan, with a particular focus on structural biology and human health. And I'll start today with a disclaimer. There's only so many ways that you can introduce a talk about cilia. So the first 4 slides are almost identical to Alan Brown's yesterday. I promise today we'll start to show you some new stuff afterwards. So yeah, cilia are these hair-like organelles with diverse functions. We see them in multiciliated epithelial cells. So these have hundreds of cilia on the surface to create fluid flow in the lungs and in the fallopian tubes and in regions of the brain. We also see motile cilia in sperm cells, and these create propulsive force. The same idea is seen in Chlamydomonas, which is our model organism. So all the work that I'll show you today is done in clammy. These have 2 cilia, which again help the cell swim through liquid. And then, of course, more or less every mammalian cell has a primary cilium. So these do not have any of the motors, but they have a large concentration of different signaling molecules, and they act as a sort of cellular antenna during cell development and maintenance. And so cilia are linked to a large number of human pathologies. So when you have a mutation in a cilia or cilia-related protein, you get all of these different phenotypes affecting all different sorts of different regions of the body. And so it's really important that we understand exactly how cilia function, and then we can understand exactly what's going wrong in these different diseases. And so we are now familiar with the structure of the motile cilium. They're built around this microtubule cytoskeleton. Microtubules are polar filaments, and the minus ends of microtubules and cilia are focused on the cytoplasmic basal body, and then they project up to 10 microns away to the tip with the plus end. And if you look through the cross-section, we see this very well-defined axoneme structure. We have 9 microtubule doublets in a ring with all these different outer dynein arms and inner dynein arms and radial spokes. And so the question that I want to sort of get to today is, how are cilia assembled? So how do you get to this structure? And the fundamental thing that we need to consider when we think about cilia assembly is the fact that there is a diffusion barrier between the basal body and the cilium. So this is in the transition zone in this sort of region here. So this means that all of the proteins in the axoneme and all the other proteins which you find in cilia need to be selectively actively transported from the cytoplasm to the tip of the cilium for incorporation. And we can see evidence of this transport mechanism when we look in cryo-electron tomograms of Chlamydomonas cilia. So this is one that I took. And as we can scroll through, we can see all these different microtubules and all the different axoneme components. If we stop here, we can see that there are these bulges in the membrane with these dense polymeric structures in between. And these are called intraflagellar transport trains. And the role of these is to bind all of these different cargoes, which need to be taken to the tip to help the cilium grow, as well as molecular motors. So they're essentially a scaffold and an adapter between the motors and the cargo. And then when we look in live cells, we see it's a very dynamic process. So this is one of these IFT proteins, which we've labeled with a fluorescent tag. We can see that there's rapid unidirectional transport from the base to the tip. And then that's actually a new process going from the tip back to the base. So it's occurring in cycles. And when you don't have intraflagellar transport, you essentially don't get a cilium. So this is a comparison of a wild type in plastic EM, wild type cilium, and a mutant where just one of these IFT proteins is mutated, and you don't get any cilium, and obviously you don't get any functionality. So what do we know about these IFT trains? We know that they consist of polymers of the IFT-A and IFT-B protein complexes. And together, these complexes are made up of 22 different subunits. And as we saw in the live cell imaging, these IFT proteins can be thought to go in a cycle throughout the cilium. So if we start off in the cytoplasm, we have these individual complexes. And then at the basal body, so this is the cytoplasmic base of the cilium, we have polymerization first of IFTB, and this then recruits IFTA. So this is based on some really nice fib milling and cryo-ET that was done in collaboration with Ben Engel's group and Gaia's group. And in these images, we can see that the IFTB in blue is polymerizing much longer in the initial stages of assembly than IFTA. At this stage, the trains are binding to the cargoes and the motors. And when we look inside the actual cilium, we see a mature anterograde train. So this has IFTB and IFTA at the same sort of length as each other. And these trains are carried by kinesin to the tip, where the cargoes are unloaded and incorporated into the axoneme structure, at which point the trains undergo a remodeling process. So it's not quite clear exactly what happens here. The IFTA and the IFTB could completely disassemble from each other, or the anterograde train structure could undergo some form of internal remodeling. What we do know is that when we see the retrograde train, so these are the trains which go from the tip to the cytoplasm again, carrying a new selection of cargoes, which are either targeted for recycling or degradation in the cytoplasm, or for signaling roles in primary cilia, for example. When we look at these retrograde trains in our tomograms, they are a completely different structure. So they have much lower electron density, they're much more elongated, despite being made up of the exact same IFTA and IFTB complexes. And then back in the cytoplasm, the retrograde trains disassemble and we start the cycle again. So it's a really intriguing process where you have the exact same constituents, the IFTA and IFTB complexes, binding to different proteins, engaging in different interactions and forming these different structures, but we really don't understand the sort of mechanistic processes allowing this to happen. So we know a reasonable amount about the IFT complexes in an isolated sense. So based on a couple of decades worth of in vitro biochemical studies, pull downs and so on, we have this nice network of interactions where we can know exactly, or broadly speaking, which IFT proteins are interacting with the others. But from a structural perspective, there is only limited information, limited to some fragments of co-crystal structures and this sort of thing. So we don't fully understand how these complexes are formed. And then at the other end of the scale, from the in situ data, a few years ago, Marika Jordan in Gaia's lab made these subtomogram averages of anterograde trains in CLAMI. So the resolution was 37 or 33 to 37 angstrom. So, I mean, these were really informative. They showed us which regions of the train corresponded to the IFTA complex and the IFTB and so on. But obviously, we're not able to get any sort of internal conclusions from this resolution. So this leaves us with sort of a few questions, which I can summarize here, which I want to address in the talk today. So in a sort of hierarchical fashion, most basically, what is the structure of IFTA and IFTB? How do these different subunits interact with each other to form these complexes? And then moving up, how do these complexes interact and form lateral interactions to form the polymers? And then finally, how do the IFTA and IFTB polymers interact with each other to form the mature anterograde trains that we see in cilia? Okay, so to address these questions, I wanted to revisit the subtomogram averaging of anterograde trains in CLAMI. And so a few years ago, Marika's averages were using PEAT. And since then, we've got a whole host of new softwares and technologies which we can use to really push the resolution. So CLAMI are fairly straightforward to do cryo-ET on. You just apply your solution to the grid of cells and plunge freeze them, and they stick their cilia into the holes quite happily. We can then select our regions manually and start to collect our tomograms. And so the diameter of these cilia is 250 to 300 nanometers. So it's getting onto the thick side, but it's thin enough that we don't need to do any fib milling or any additional thinning. So I collected 600 new tilt series on the Cryos at the Human Technopole using fairly standard data collection conditions, and moved on to the new processing workflows that we wanted to use. So after reconstructing our tomograms, I realized that there was no way to do template matching with this sample. I was just picking all sorts of rubbish inside the axoneme. So I have to manually pick all of my particles. So I go through each of the tomograms and I identify the anterograde trains. I pick a line through the train as a contour, and then I oversample to generate my particles, with the idea being that you then, after refinements, you can remove any duplicate or neighboring particles to get to your nice refined particle coordinates. So when I move forwards with these particles in the warp and Reliant and M pipeline, essentially when we did global refinements, we don't refine any signal. So if this is a cross-sectional view on the left here, we see that there are the same features on the bottom as on the top, which is indicating that it's not differentiating between the top and the bottom of the complex, which we know looks different. And on this top view, we see that there's not very much signal being refined outside of the mask. And considering that it's a long filament, we know this is indicating that you're only really aligning noise. So we took a step back, and I think the best way to now proceed with this sort of data is to work out a way to increase the amount of signal in our subtomograms, because this is the conclusion we came to. The IFT B and IFTA complexes are quite flexible, quite floppy, so they need a real boost of signal before we can start to refine them in Reliant. And of course, the warp Reliant M pipeline has a way of boosting signal. So the idea is that you would extract your subtomograms in warp, you'd get a 3D refinement in Reliant, and then in M, you would do your refinements of your tilt series alignments. So if you're not familiar, this is broadly similar to particle polishing in single particle workflows, where you go back to your individual subtomograms, your individual particles, and work out the maximum or the best way to align all of the tilt series to maximize your signal in your particle. And this generates a local sort of map of the optimized tilt alignments throughout your tomogram. And so we couldn't use our IFTB samples for this because they don't refine. But what we are able to do is find a nice sort of fiducial model in our axoneme. So I went through our data again and picked the central pair microtubules. We can actually do this with the Cryolo filament tracker now quite nicely. These are ones which we see in the center of the axoneme, and which Alan showed you yesterday, along with Kai Zhang also having a nice single particle structure. These are very high density and repetitive structures that can be refined to very high resolution. So they seem like a good sort of candidate to bootstrap our tilt series refinements. And indeed, when we do global refinement with our initial tilt series alignments, we are able to refine this filament. We got to 18 angstrom resolution with 20,000 or so subtomograms. But then the real improvement came after we used the M tilt refinements on this. So now we're getting to 8 angstrom resolution without fully unbinning this data. So we've improved the amount of signal in our particles to give us a 10 angstrom at least improvement in resolution. So we can now use these optimized tilt alignments to go back to our IFT trains. This is what we had before optimizing our alignments. And now this is what we have after. We can see that we are aligning signal quite nicely. We see other features outside of the mask and so on. And so in the end, I ended up with 3 new subtomogram averages. I masked 2 different regions in IFTB to 10 and 11.5 angstrom resolution. And then the IFTA complex went to 18.5 angstrom resolution, so it's a bit more flexible. Okay, so with these new structures, we can start to answer these questions. So firstly, what can we say about the internal structure of IFTA and IFTB? So of course, at 10 to 12 angstrom resolution, we can't do de novo model building. So we started off with a selection of AlphaFold models. And we informed the way that we would ask AlphaFold to predict these structures based on these biochemical interactions. So for example, we know that IFT54 and IFT20 are a coil that interact as a heterodimer. So we folded the multimers in this way, and then we can start to fit them into our density. So the first protein that we fit was IFT8852. IFT88 is a TPR superhelix. So this density was quite obvious just from visual inspection. We could fit this minor complex into the density. We used NAM-donators, molecular dynamics flexible fitting, to fully refine it into our density. And then because we know that IFT88 interacts with IFT70, for example, we can then fit this into the neighboring density, use the NAM-donator again to fit it in properly. And using the combination of the features that we see in our map, as well as the biochemical interactions, we were able to build up a model of the entire train. And yeah, so we repeated this exact same process with the IFTA complex. We now have this complete model of the anterograde IFT train. We know exactly what each protein is interacting with and so on. I won't go into too much detail about this sort of nitty-gritty at the moment, but what I will say is that shortly after our subtomogram average preprint came out, there were 3 or 4 different single particle structures of IFTA also released. So this is my structure here, and these are the 3 high-resolution single particle structures. I mean, firstly, we can see that the structures are very similar. So this validates the fact that my low resolution method was at least on the right track. But what we can also see is that these are all from different organisms. So my structure is from clammy. We have 2 structures from human. Alan Brown kept this in his back pocket yesterday, but he has a structure from leishmania. And the fact that all these structures are the same, or near enough the same, indicates that what we're seeing is a really highly conserved process from our nice model organisms in unicellular protists all the way up to humans. Okay, so I'll start to answer this next question now. So how do these structures polymerize? And what I'm showing you here is 3 repeats of the IFTB filament. And I'm showing you the central repeat colored by subunit. But now I'm going to change the coloring to the AlphaFold PLDT score. So this is the estimate that AlphaFold gives you on a per residue basis of its confidence of the conformation in each residue. So by and large, we can take this as an estimate of the flexibility in these different regions. And so blue would be lower confidence, higher flexibility, and red is vice versa. And so I think what we realized is that the lateral interfaces tended to have the highest amount of flexibility. So these regions here, this region here, and this region here. And this is unusual because it's not sort of polymerizing in a ball and socket sort of configuration. It's undergoing a flexible to ordered transition, especially considering there are like-to-like interactions, so 2 flexible regions interacting with each other. And when we look closer at IFT172, which is one of these lateral interactions, we see that it undergoes large-scale conformational changes. So this is the amount of movement to go from our original AlphaFold prediction to our refined model. So again, we're undergoing the flexible to ordered transition, and we're undergoing large-scale conformational changes. And given that there's no ATPase or anything in the IFT proteins, it seems an odd way to polymerize, or it seems like this may be perhaps energetically unfavorable. So what we're starting to think about how this might work is through an insight at the basal body. So this is the mature anterograde train structure that I've shown you now from the middle of the cilia. And this is a subtomogram average from Ben Engel from the basal body, so the same data set that I showed you earlier. And in the assembling anterograde train at the basal body, we see this extra density underneath the train. And it's not clear what this is. So what we're starting to think as a potential model is that this is an exogenous packaging factor. So it's something which is helping the IFTB polymerize into these conformations. And this could add a level of regulation to the assembly. It can mean that you only get assembly at the basal body and not at the tip or in the cytoplasm where you don't want it. And then one more fanciful theory that we're working on is that this is pushing the anterograde train into a sort of high energy state, which then through a trigger at the tip causes the train to maybe release its tension and induce its remodeling into a lower energy retrograde state. But this is something that we can only really start to think about if we have a structure of the retrograde train. Okay, so that is sort of what we're thinking about with regards to polymerization. And then the last question I want to come to is, how do IFTA and IFTB polymers interact with each other? So the problem with answering this question is that IFTA and IFTB in these trains have a mismatch in periodicity. So the IFTA average, or the IFTA repeat, is 11.5 nanometers, and the IFTB repeat is 6 nanometers. Because these numbers are not directly divisible by each other, it means that in IFTA, we have the region corresponding to IFTB blurred out, and vice versa. So we were able to overcome this with 3D classification. So I did 3D classification of the region corresponding to IFTA in our IFTB average. And this shows us short regions where the IFTA is in a set register relative to IFTB. And when we do this, we see that there is an extra density emanating out of IFTB and contacting IFTA that we didn't see before. So this density in dark blue. And when we compare different classes, we see that these sort of tubular densities are very flexible. So they're able to keep IFTA and IFTB tethered to each other in a wide range of conformations. So we wanted to think about what the identity of these densities could be. And in our model, we know that IFT8174 is the protein in this region. And if we compare what we model in this model to the left versus the original AlphaFold model, we see that there are 4 extra coil segments, which we assume to be flexible in our overall model. When we look back at our density and fit everything in, this extra density corresponds very nicely in size and shape and location to the 5th coil of IFT8174. And because it's a segmented coil, it has these hinges between coil segments. So this really allows it to adopt all these different conformations to keep IFTA on top of IFTB. So additionally, we saw a second extra link between IFTA and IFTB after classification. This is in the center of the train, so I'm showing you it now. It's the green one coming up from IFTB. It sort of projects up into the center of IFTA. And when we actually look at it more closely, we see that it cross-links, or it bridges 2 adjacent IFTA proteins above it. So what we think this could actually be doing is aiding the polymerization of IFTA on top of IFTB and stabilizing the interaction to help it polymerize. So again, we thought about what this could be. And again, in this region, there is a large unmodeled and flexibly bound domain. So in this case, this is the modeled region of IFT172 versus the original AlphaFold prediction. We have this big C-terminal domain, a nice flexible linker region here. And when we look in our density, again, it corresponds very nicely to the density that we see. So again, this is what we think is going on to keep IFTA and IFTB tethered. So yeah, so we think these 2 flexible interactions that we see at the most basic level keep IFTA and IFTB attached to each other. The fact that they are flexible means that it creates, or it allows IFTA and IFTB to remain tethered in the flexible and dynamic environment of beating motile cilia. And then finally, we think this mode of interaction can help with assembly. So we have 2 sort of tentacles searching out on this IFTB train at the basal body, and it can recruit IFTA and help it polymerize on top of the IFTB train. So that brings me to the end. So today I've shown you how we used subtomogram averaging and AlphaFold predictions to generate the first models of IFT complexes in IFT trains. We've shown how these complexes are organized, and we provided insights into how they may assemble. Obviously, we need more experiments to really show us exactly what's going on. And then in regards to the questions that I haven't answered, obviously these structures that we see are just the sort of adapters, And we don't see any cargoes in our overall averages because they're all completely heterogeneous and they just average out. So this is something we need to search for in the future. And of course, this is only a snapshot of the cycle of the complexes that I showed you at the start. So we really need to understand exactly what's going on in the retrograde trains before we can start to understand how these complexes are fully functional. And so with that, I'd like to thank Gaia and the Juno group. Helen Foster did some of the AlphaFold predictions. And I'd like to thank the HT-EM facility and the entire structural biology department. We started in 2021 and we keep on growing. And I think there's going to be some really cool stuff coming out of this new department soon. So you should watch out. And thank you all for listening.